In this video, the focus is the transport of proteins into the nucleus. Before we start looking at the mechanisms, let's quickly review some things that I think you already know about the nucleus. The nucleus is one of the most recognizable structures in the cell. In this cartoon of an animal cell, you can look at it and know that the blue structure is the nucleus. And if we zoom in even further and look at this cartoon, we can see some of the structures that make it so identifiable. The squiggly lines, that represents the chromatin, the DNA. Unless a cell is actively going through mitosis or meiosis, the chromosomes won't be condensed. Instead, the DNA is unwound, allowing access for transcription factors to come in and express the genes. The dark spot here is the nucleolus. It's involved in making ribosomes and ribosomal RNAs. Those will have to be exported then to the cytoplasm in order to do their work. Another feature of the nucleus is that it has a double membrane structure. There's an inner nuclear membrane, this is a full lipid bilayer, and an outer nuclear membrane, another full lipid bilayer. I want you to notice a couple things. First is that the outer nuclear membrane is continuous with the endoplasmic reticulum. You can probably identify the rough endoplasmic reticulum by all the dots, which represent ribosomes, and see how it's continuous with the outer nuclear membrane. The other thing I want you to notice is that there are places where the inner nuclear membrane and the outer nuclear membrane fuse. And where that happens, there are holes in the nuclear envelope. We call these the nuclear pores. And these are going to be the structures through which messenger RNA is going to leave the nucleus and proteins are going to enter the nucleus. Now in a cartoon like this, the nuclear pores look really big, but actually they're small structures, and to see them we would have to use electron microscopy. That's what this picture is showing you here. I know that it's a little intimidating to look at electron micrographs if you're not used to them. Fortunately, the site where I got this image also colors them for us. So here on the right is the same image, but now it's been color-coded for us. I want you to see that purpley structure. That's the nucleus, and the especially dense spots, that's where the chromatin is. In this picture, mitochondria are colored red. The cytoplasm is colored green. The light blue, that's the endoplasmic reticulum. And can you see the purple space that lies between the nucleus and the cytoplasm? That's the perinuclear space, the space that's lying between those two lipid bilayers. Let's zoom in a little bit more and see if we can see a nuclear pore. This structure here is a nuclear pore, and if you look on either side of it, you can see how the inner and the outer nuclear membrane have come together. The nuclear pore is going to allow access between the nucleus and the cytoplasm. This is different from another structure. Over here we can see that there's continuity between the ER lumen and the perinuclear space. But that's a different connection than the nuclear pore, which is linking the cytoplasm and the nucleus. To illustrate how proteins are imported into the nucleus, I'm going to be using this cartoon. The nuclear pores here are represented by the structures that look a little bit like a basketball net. You might be wondering why the nuclear pore is drawn in such a way, but the answer is because this is what we really think they look like. Here are some scanning electron micrographs showing first the cytoplasmic surface. So if we were walking on the outside of the nuclear envelope, what would we see? And you can see that we would see these ring-like structures with some strands coming off them. That's what's indicated by the dark blue part of the nuclear pore. On the other hand, if you were inside the nucleus and you were walking along and looking at the underside, you would see these basket-like structures. The reason the biological illustrator has drawn them looking like little baskets is because they look like little baskets with nets. The nuclear pores are going to represent fluid-filled pores through which material can, in theory, move between the cytoplasm and the nucleus. It's hard to tell from these cartoons just how large they are and what kinds of molecules can pass. This question's been addressed using some experimental techniques. It's possible to take dyes of known sizes and add them just to the cytoplasmic side and then wait and see if they can pass into the nucleus. If this is done with small molecules, then they'll freely diffuse back and forth. Experiments like this have shown that any molecule that's under 5,000 Daltons can freely diffuse. And just for a sense of how large this is, ATP, that's about 500 Daltons, so this would be 10 times the size of an ATP molecule. Can all molecules move through the pore? No, they can't. We know this because if you take a larger dye molecule, like these here, they won't pass through. And the absolute limit seems to be about 60,000 Daltons. A molecule that's bigger than 60 kD cannot enter the nucleus from the cytoplasm by diffusion. But here's the tricky thing. We know that most proteins are actually 60,000 kilodaltons or larger, and many proteins do work inside the nucleus and must enter from the cytoplasm in order to get in there. For example, the estrogen receptor, that's about 60 kilodaltons. It works in the nucleus. There must be a way for it to get there. Here's another example, the BRCA1 gene, that's the breast cancer gene. This is involved in DNA repair. It's critical to have it in the nucleus where it can repair damaged DNA, but it is 200 kilodaltons, 200,000 Daltons. That's way too big to enter by diffusion. There must be then some kind of active transport system that will allow these large proteins to enter into the nucleus. So how do nuclear proteins get into the nucleus? Nuclear proteins are post-translationally sorted. They're synthesized in the cytoplasm on ribosomes. When synthesis is finished, the protein will fall off. Proteins destined for the nucleus contain what's called the nuclear localization sequence. Notice how in this cartoon I've drawn it somewhere in the middle of the protein. Unlike some other signaling sequences that we've seen, it doesn't just have to be at the N-terminal. We could find it anywhere in the protein. There's a family of at least 20 proteins that's involved in moving nuclear proteins from the cytoplasm into the nucleus. We'll call these importans. In one of the best studied examples, two subunits are required. The important alpha subunit binds to and recognizes a protein at the nuclear localization sequence. This then recruits important beta to join the complex. And once they're together, the important beta, which has a high affinity for proteins lining the nuclear pore, is able to move that protein into the nucleus. Now that the nuclear protein's inside the nucleus, we need to get it to be released from the importans. This process is accomplished with the help of a protein called RAN. RAN is a member of a family of proteins called small G proteins. What they have in common is that they can exist in two states, and it will depend on whether they're bound to GTP or GDP. RAN GTP is found primarily in the nucleus, and it's going to act as the trigger to release the nuclear protein. What happens is the RAN is recruited and binds to the important beta subunit. Once this happens, the alpha subunit loses its affinity for the nuclear protein and will also be released. This allows the protein now to get to work on the nucleus. The next step is to recycle the importans and return them to the cytoplasm so that this process can be repeated with a new nuclear protein. Once the beta subunit has bound the RAN GTP, it will translocate and be moved out of the nucleus and into the cytoplasm. The alpha subunit requires a helper protein. It needs to bind what's called an exportin, or an export factor, as well as a GTP molecule. And once these are associated, they now will leave the nucleus and also exit into the cytoplasm. In order for the alpha and the beta subunits to be useful, we need to get them to release from the GTP. This can happen if we can get the RAN molecule to switch to the other state and be bound to a GDP instead of a GTP. Well, RAN has a low intrinsic GTPase activity, but it's slow. So to speed it up, what happens is that there's a molecule called RAN-GAP, GTPase activating protein. That molecule, which normally is present in the cytoplasm, can associate with the RAN molecule, and when it does this, it speeds up the activity of the RAN molecule. It will now hydrolyze the GTP molecule, and as it does this, it will release a phosphate group and be left bound to a GDP. In this configuration, it loses the affinity for the beta important, and now that molecule is free to be recycled. In a similar way, RAN-GAP can associate with the RAN GTP that's associated with the alpha subunit, activate it so it also hydrolyzes GTP to a GDP, at which point the alpha subunit will be released. Now the alpha and the beta importance subunits are available to move the next protein that has a nuclear localization sequence into the membrane. The RAN gap will release its molecule and float away, ready for use the next time a RAN GTP is present. And now we just have to return the RAN GDP to the nucleus and restore it to the other state, the RAN GTP. This is accomplished with a protein called NTF2, nuclear transport factor 2. That protein in the cytoplasm has the property that it can bind RAN when it's bound to GDP, and then move those molecules back into the nucleus. Once the RAN molecule has been returned to the nucleus, we need to reset it by having it be released from the nuclear transport factor and also by having it replace the GDP with a GTP. This is accomplished with the help of a protein called RAN-GAF, shown here in purple, a molecule that's normally bound to the chromatin in the DNA. GEF stands for guanine exchange factor. So this molecule can bind RAN, and when it does this, it can do a couple of things. First, NTF2 will be released from the complex. The second thing is the exchange of the nucleotide. Once associated with the RAN-GEF, the RAN molecule will drop the GDP molecule and pick up a GTP molecule instead. It can now dissociate from the RAN-GEF molecule, and we have the RAN with the GTP bound to it, ready to be used for another cycle of importing and exporting proteins. Let me point out a few more things before closing out this video. The first is that we see in cells that there's a high level of RAN GTP in the nucleus and a high level of RAN GDP in the cytoplasm. This has to do with the relative concentrations of the RAN-GEF, RAN-GEF, in the nucleus and the RAN-GAP in the cytoplasm. The next thing I want you to notice is that at no point in this cycle was the nuclear localization sequence cleaved from the nuclear protein. This is different from some other signals that are one and done. They bring the protein to a target in the cell and then it's clipped off. This doesn't happen with the nucleus, and what it means is that nuclear proteins might go through repeated cycles of import and export. In fact, many proteins contain both a nuclear localization sequence and a nuclear export sequence. And if a exportin, a molecule that can bind to the protein and move it out of the cell, binds to the nuclear export signal, the protein will be moved back to the cytoplasm. If the protein in the cytoplasm has the nuclear localization sequence exposed, it can be moved into the nucleus. And many proteins that cycle back and forth between the cytoplasm and the nucleus are associated with other proteins that can mask either the nuclear localization or the nuclear export signal in order to keep them in the compartment where they're needed. If any part of this process breaks down and the cell is unable to move proteins from the cytoplasm to the nucleus or vice versa, it can be devastating, and deficits in this process are implicated in a number of diseases, such as cancer.